Hello and welcome everybody to our chapter 23, which is where we will begin discussing the urinary system. So for this system, the composition is fairly simple. We only have six principal organs, six main organs here at play. Two kidneys, so paired kidneys, two ureters, those are the tubes connecting your kidneys down to your bladder. And then from the bladder out, we have the urethra. So here we can see those organs. You can orient yourself. Here's our diaphragm. We are solidly in the abdominal cavity. We've removed a lot of our abdominal organs to get this view, but we can see our paired kidneys, one here, and you can even see here that this left kidney sits just a little bit higher than the right kidney, which is one way that's kind of an easy diagnostic if you're looking in the abdominal cavity. Here's our posterior view. So here is our right kidney, that little bit lower. Here is the left. And you can see they sit pretty much right under the 11th and 12th costals, the ribs. Then we have our paired tubes, these little guys right here, which will drain all the way down into our urinary bladder, which sits right in the pelvis. And this is a female diagram, so you can see the urethra is rather short right there. If this were a male, the urethra would be much longer, and it would connect to some of the tubules and glands like the ductus deferens, the prostate, everything coming from the testicles would connect, and then the outflow. So when it comes to functions of the kidneys, right, we have to think about waste. There are a lot of functions of the kidneys, but a big one is waste products. Okay, the kidneys are filters. They will filter your blood of any metabolic wastes. So that's things like urea, uric acid, anything that is produced through metabolic processes that we don't want to accumulate in our body. Now, again, not the only function. We also see a lot of regulation take place in the kidneys as far as blood volume. Blood volume is mainly dependent on presence of water. How much water do you have in your blood? Well, we can excrete water via the kidneys, but we can also reabsorb water through the kidneys. So we can pretty successfully change our blood volume based off of how much water we absorb or secrete. We can also change the pressure of the blood through blood volume and osmolarity by the components, the concentration of big proteins like albumin, for example. Now, through that regulation, we also see the kidneys play a big role in acid-base balance. Remember, when it comes to acid-base, it's big on components like carbon dioxide, carbonic acid, H+ concentration. We're going to see a lot of that here again in the kidneys. We also see a role in blood cell creation. Specifically, the kidneys will secrete erythropoietin. And if you remember, poiesis means creation. Erythro is referring to red blood cells. So erythropoietin, when secreted by the kidneys, will stimulate production of red blood cells in our red bone marrow. Now, we can also regulate calcium by specifically regulating calcitrol synthesis in the kidneys. We can help clear hormones, some of those chemical messengers, again, detoxification, and even the kidneys even play a role in starvation. So when you starve, when your body is deficient of nutrients, they can actually create energy. They can create glucose from amino acids. So they will take the building blocks of proteins and turn it into sugar. So if we're talking about filtering waste, we have to actually define what waste is. Waste is anything that is useless or that we have too much of. Anything that our body is not going to use. So we have to get rid of it. But we can get more specific. There are things called metabolic wastes. Okay, metabolic is referring to metabolism. That is the building up and breaking down of molecules in your body to maintain energy homeostasis. So anything that is produced as a product of that metabolism that we don't need or use is called a metabolic waste. For example, carbon dioxide is a metabolic waste because it is produced through cellular respiration. So when we break down a nutrient like glucose, turn it into ATP, cellular energy, we make a little bit of carbon dioxide that has to be excreted. Now, among wastes, the most toxic by far are the wastes that contain nitrogen. And we literally call them nitrogenous wastes. And here's a few examples. Urea, okay, from protein catabolism. Okay, catabolism, think back 1306, okay, that's breaking down proteins. So whenever you break down proteins, you produce urea, okay, which is a nitrogenous waste. Uric acid is another. Okay, this time it's when you break down nucleic acids. So if you're breaking down DNA or RNA, you'll produce a little bit of uric acid. All right, then we have creatinine. Okay, this is a waste product of creatine phosphate catabolism. So when we break down creatine phosphate, we make creatinine. Now, blood urea nitrogen level. Okay, this is something that we can measure, okay, BUN. This measurement is essentially how much nitrogenous wastes do you have in your blood. This can be an indicator of kidney function. If you have a normal concentration, a normal BUN, it's about 10 to 20 milligrams per deciliter. However, if your kidneys aren't working well, that means nitrogenous wastes are not getting released from the body properly. They build up, and it can elevate this BUN, this blood urea nitrogen. This is called azotemia. Now, we also have uremia, another condition involving the kidneys here. This is a syndrome of diarrhea, vomiting, dyspnea, or cardiac arrhythmia. Okay, so this is usually the effect, but the reason why is because you have too many nitrogenous wastes. So maybe you have azotemia, an elevated BUN, and that's leading to uremia, where you're going to see diarrhea, vomiting, dyspnea, all of these because of those toxic levels of nitrogenous waste. Now, what you have to do, you either have to give them a new kidney that works well, okay? Give them something that's going to filter their blood properly, or hemo means blood, dialysis is referring to filtering. So hemodialysis is literally filtering the blood. So we can remove the blood from the body, we can filter it, and then we can give it back. Essentially, a machine will be doing the work of the kidneys. So I just wanted to show you a picture of these major nitrogenous wastes. Urea, uric acid, ammonia is another, and then our creatinine. Now, again, I'll just highlight the reason why we call them nitrogenous wastes, all of that nitrogen. So how do we get rid of nitrogenous wastes? Well, that process is known as excretion. Anything that is going to separate those wastes and eliminate them. And it is not just the urinary system that is going to do this. We have a lot of ways to excrete waste, and for good reason. We really don't want that waste to build up and kill us. So our lungs can do it. We've already talked about this, right? CO2 is a metabolic waste product. So whenever you take a nice deep exhale, you are expelling waste. You're excreting waste, a CO2 product. So small amounts of gases, sometimes water, can be excreted through the respiratory system. Your integument, your skin, okay, can also excrete waste. in sweat. So your sweat contains water, but it also contains salts, inorganic salts. If you've ever had a pet that likes to lick you after you sweat a lot, it's usually because they like the salt in your sweat. They like that salty taste. Lactate, urea, okay, one of our nitrogenous wastes, can be found in sweat. Our digestive system, which we should be very familiar with as per our last unit, we can get rid of a ton of stuff: water, salts, carbon dioxide, lipids, bile, right? A lot of our bile we reuse, but the pigments can be excreted. Bilirubin, for example, cholesterol, other tons of stuff. Urinary system. This is another big one, especially for metabolic wastes. So we're going to see a lot of urea. We're going to see a lot of uric acid, ammonia excreted through this system. We also see toxins, drugs, hormones, salts, H+. Right, this is how we're going to regulate our acid-base balance. It's big here. Okay, H+. We can retain it if we need to be more acidic. We can get rid of it if we need to be more basic. So positioning, we already saw a hint of earlier, but they lay on the back of your abdomen. If you compare it to the spine, they sit right at T12, so the thoracic spine to the lower lumbar spine, L3. And they don't sit evenly. The right kidney is lower. And think back to the lungs. All right, anytime we see differences in size or positioning of paired organs, typically it's because of the presence of another organ getting in the way. So in this case, the right kidney sits lower because that giant right lobe of the liver is sitting in the same area. So it kind of displaces that right kidney a little bit lower. Now, in terms of ribs, it is your 11th and 12th rib, specifically the 12th, that's going to do most of the covering, reaching all the way down to the middle of the left kidney. But that kidney, the top of it, will reach the 11th rib as well. It is retroperitoneal, in this case sitting behind that peritoneum on the back of the abdominal wall. So retroperitoneal, the ureters are also behind that peritoneum, same urinary bladder, renal artery, vein, adrenal glands, all considered retroperitoneal. All right, so we're taking a cross section here so you can essentially see exactly where these kidneys are laying retroperitoneal. So you can see here is the majority of our abdominal cavity. This is our peritoneum lining it here. And you can see the stomach up above, some of the small intestine here, the spleen over here. And you can see that the kidneys sit behind all of that, and they are encased. They're encased in a fibrous capsule. They're encased in a layer of fascia, which is connective tissue. And they also have a fat capsule. So there is a lot of support for our kidneys. Your kidneys, as you'll see soon, are very important and highly vascularized. So we need to make sure they are protected and anchored to the tissues around them. But this is why we call them retroperitoneal. They're behind the peritoneum. For an average individual, your kidney is about 150 grams and about the size of a bar of soap. So not too huge. If you were to look at the lateral surface, it is convex and the medial is concave. So what we mean by that, the inner surface is concave and then the outer surface is convex. So we get this kind of half moon shape almost. Now, again, we got a hilum. We've seen this in other organs. It's the same thing here. Entry and exit point to the kidneys. So it's going to receive the renal nerves, blood vessels, lymphatics, and the ureter, which is going to drain the waste product out. if you look at the coverings of our kidneys, there's a lot going on. Okay, I mentioned them a few on the last slide. One, the renal fascia. This is deep to the peritoneum. Okay, this is going to bind the kidney to the abdominal wall. And again, fascia is a type of connective tissue. So by the name alone, what's it going to do? It is going to connect. It's going to anchor and support our kidney to the abdomen itself. Then we have our fat capsule. The fat capsule, just like you might think, is all about insulation and protection. We're trying to cushion this kidney and hold it into place. Again, there's a lot of vascular connection here, so we do not want our kidneys to be moving around. We don't want them to be damaged, so we need to make sure that we anchor them and we cushion them. Now, lastly, we have the fibrous capsule. This is going to surround your kidney in its entirety and protect it from trauma, infection. We mostly see collagen in this layer, so a lot of structural support that connects both the fibrous capsule to the renal fascia, essentially, again, anchoring and protecting our kidney. Now let's look inside the kidney itself. So your kidney is made up of renal parenchyma, which is a glandular tissue. This is going to contain the cells that are going to actually form urine. Now, again, we got a C shape to our kidneys. Okay, that's that kind of half moon that I said earlier, this convex and concave portions. Now, when we are talking kidneys, you are going to see what's called a renal sinus, which is essentially a cavity in the middle of your kidney that's going to contain blood, lymphatic, nerve, urine collecting structures, all of it. Now, we also divide our parenchyma, our tissue of our kidney, into essentially two distinct regions. If we are toward the outside of the kidney, okay, we call it the renal cortex. If we are toward the inside of the kidney, we call it the renal medulla. Okay, and this would be if I, cut my kidney in half. We're looking at it here. I'll show you a better picture of this later on, but just two different regions. Now, the boundary that separates them, where they touch right here in the middle, okay, is called the corticomedullary junction. And again, cortico refers to the cortex, medullary means medulla, and junction just says, hey, this is the point where they meet. Now, we also see what's called renal columns. So essentially, this cortex, this outer portion, will extend inward into the medulla toward the sinus, which sits at the very center. Okay, so they will have these extensions inward. They're called renal columns, and they divide our medulla into these pyramids. So the pyramids are just these spaces between the renal columns, called renal pyramids. All right, so we then divide our kidney into lobes based off of these columns and pyramids. So the lobe of a kidney, or one lobe of a kidney, consists of one pyramid and its overlying cortex. So it would be one of these regions. Okay. And again, I got a better picture on this later on. We also have a series of drainages within this. The minor calyx, you're going to see at the tip of each pyramid that we made in the medulla, there's what's called the papilla. It collects urine from the cells that are creating it. Then we have the major calyx. So when you have 2 or 3 of these minor calyxes converge, we call it a major calyx. It drains into the renal pelvis, which will drain into the sinus, which will then drain out into the ureter. All right, so here is finally a good picture of what I've been talking about. So let's start with, on the right we have a diagram, on the left we have the actual anatomy. So I'm going to kind of show you both at the same time. All right, this outer layer is called the cortex. So this little area on the very outside of the kidneys, cortex here. And then you can see it extends inward like so, like here. All right, and those extensions will form the pyramids of the medulla. So that's these guys right here, pyramids. And you can see one here and another one here, not as pyramidal, but okay, those are the pyramids we're talking about. Now, if you look at the very tip of that pyramid, we call this the papilla. Okay, very tip of the pyramid, that drains our waste products into the minor calyx. Okay, so each minor calyx is connected to one of these pyramids. Okay, it drains from the pyramid. So here, 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 so on and so forth. Now, when you converge two of those minor calyces, here and here, here, that becomes a major calyx. Okay, here would be a major calyx, for example, right? The convergence of these minor calyces. Then, when you converge multiple of these major calyx, so here's another major calyx here, that convergence, we get the renal pelvis. That is this area right here. Okay, renal pelvis. And that renal pelvis is going to drain into a ureter. Same thing here. We're going to see this one come down. We're going to see this major calyx here drain down. And then this is officially our renal pelvis, which will come into the ureter. Now, the hilum region, again, this is that entry and exit point. So all of this is considered the hilum region of the kidney. Same thing here, all of this major entry and exit points. And then the last thing to mention is the renal sinus. So this is, again, that kind of innermost portion of the kidney. These little areas right in here, between everything. Right there and there. And then if we look on our cadaver, that's these areas. Okay, basically just these empty pockets that are great for vascular connection, but they sit in the very, very center of the kidney. Now, why did we need so much protection and cushioning and insulation? Boom. Okay, your kidneys have probably the most extensive blood supply of any organ by weight. All right, they're only 0.4% of your body weight, but they receive 21% of your cardiac output. That's more than the brain even receives. We call that the renal fraction, that percentage that your kidneys receive. Now, the reason why, right? The kidneys are a filter. Specifically a filter for the blood, okay, removing those nitrogenous wastes, removing the metabolic wastes. So we need to pass most of our blood through the kidneys in order to remove waste. Now that leads us to some pretty interesting circulation pathways here. Specifically, the interesting thing about renal circulation, we are going to see two capillary beds. Yes, two different capillary beds in your kidneys. So, what I mean by that, what we've seen prior to this, right? We've seen an artery, okay, then of course you get smaller, arteriole, but we'll just keep it simple, artery side. We see a capillary bed where gas exchange occurs, and then we see the venous side. okay, venules and upward. All right, that's the typical flow. Artery, deliver that oxygenated blood, gas exchange in a capillary bed, then returned through the venous side. We don't see that in the kidneys. It's a little different. We get arteries, we get the first capillary bed, we get arteries again, we get the second capillary bed, and then we get the venous side. All right, so let's look at that circulation pathway so I can show you what I mean. We start big. We start with the renal artery. Anytime you see renal, you should think kidneys. So that's our renal artery. We get a little smaller, segmental arteries, then interlobar arteries going between the lobes of the kidney, then the arcuate arteries, then even smaller, they're called cortical radiate or interlobular. Now we're going between the lobules, which are the smaller units in our kidneys. Okay, then afferent arterioles. Okay, now afferent versus efferent. We've seen it once before, and it's going to come back. Okay, it'll haunt you in the kidneys. So afferent means going toward, efferent means going away. So let me write that in. There we go. If you want to think about it like this, you can think of E for exit. So we have our afferent arterioles, and then we hit our first capillary bed. So we are here, first capillary bed, which is called the glomerulus. Then we're not done, okay? We still have to take that blood somewhere else. Okay, from the glomerulus, we hit the efferent arterioles. This is the away. Okay, so they're taking blood away from the glomerulus. And most of these arterioles lead to our second capillary bed, peritubular capillaries. Okay, so we would be here with peritubular. Some of them lead to structures called vasa recta, which we're going to see that in the picture a little bit later on, but both of these are just capillary beds, okay, areas for gas exchange. So this is our capillary bed number 2. All right, once we have hit capillary bed number 2, we can finally move into the veins. Okay, we are now returning that blood to the heart. So it's pretty much the same thing, just reverse order. All right, we get interlobular veins. These are the small ones. They go up to the arcuate veins, interlobar veins, and then renal veins. The only difference that I want to highlight here, we have segmental arteries if we're talking the arterial side, but we do not have those segmental arteries in the venous side. We go straight from interlobar to renal veins. So segmental is missing from the veins, but otherwise it's the same pathway reversed. All right, so let's take a look at that flow in our actual kidney. So here is a cross-section of our kidney, and I'm going to start arteries, work my way down, and then back. So if we're starting with the arteries, we would start with the renal artery. Okay, it would branch off, it would get a little smaller and become the segmental artery. All right, then we go segmental into the interlobar, okay, between the lobes. So we're getting between these renal pyramids here. Then we get a little smaller, and it's labeled down here, but this up here as well, this guy. Okay, that would be the arcuate. It's going around our pyramid, kind of in an arc. Okay, you can see it here as well, the arcuate. Then we get a little smaller, our smallest unit here, the cortical radiate. This is our interlobular. It goes in between the lobules, the smaller units. Okay. All right, now the reverse. We start with the smallest vein. So that would be our cortical radiate veins. Okay, let me get a better color here. Here we go. Cortical radiate interlobular veins. It drains into the arcuate vein. We go down, we get a little bit bigger. We're in the interlobar vein. Again, a little bit bigger, but this time we skip segmental. No segmental veins. We go directly from the interlobar into the renal vein. Now, here's the flow, but in flowchart form. So you can see all the way from the heart. We start with the aorta, renal artery, segmental artery. Then we got our interlobar, arcuate, radiate, afferent arteriole, first capillary bed. That's a little bit of an ugly one, but pretend that's a 1. Our efferent arteriole, our second capillary bed, either a peritubular capillary or the vasa recta, and then our venous system, cortical radiate, arcuate, interlobar, skip segmental, renal, and back to the heart. Okay, here is the area that was a little bit too hard to see in the last picture. So we're zooming in on the microcirculation here. So you can see we've just zoomed in on essentially a single unit of the kidney. So we have a pyramid here and the cortex above it. All right, so this tube system that you're seeing, okay, these are the functional units of your kidney called the nephrons. These are going to be very important for the filtering and collection of waste. So this is where we're going to see those capillary beds. So let's take a look. Again, here's our smallest unit, the cortical radiate artery. Let's stay with artery. Let me get a better color here. Okay, artery. And then you can see it's going to hit the first capillary bed. Okay, so afferent arteriole, first capillary bed, which is the glomerulus. This is pretty much the first entry point into the nephron system. This is going to be where a lot of the filtration of the blood takes place. So afferent, then we get the efferent, okay, which moves that blood out, and then we will hit, in this case, a peritubular capillary. And literally, peritubular means around the tubes. And look where it's sitting, right? This capillary bed is literally sitting around the tubes of our nephron. And you can see gas exchange, then we hit the venous side of the capillary bed. We drain into our cortical radiate vein. Now, if we're going over here and we have a slightly different kind of nephron, okay, this is called a cortical nephron over what we just talked about, and that's because it sits in the cortex. Okay. We also have what's called juxtamedullary nephrons, and they sit between the cortex and the medulla. So, similar though. All right, we will still see our cortical radiate artery bring things in to the glomerulus. That's our first capillary bed. All right, so that's the afferent arteriole glomerulus, then efferent arteriole. But this time, instead of going around the tube system, we're forming a capillary bed called the vasa recta, which sits around this very specific loop. Okay, this is the loop over here. It's called the loop of Henle, and we're gonna talk about that in much more detail later. But the vasa recta sits specifically around that loop. But it's the same thing as far as function. It's a capillary bed. So we're going to see gas exchange, arterial to venous, and now that venous blood will drain back into our cortical radiate veins and outward. Same image here, just zoomed in for your viewing pleasure. So let's focus in on the nephron. Again, the nephron is essentially the functional unit of your kidney. This is where the filtration and reabsorption is going to take place. Now, each of your kidneys has about 1.2 million nephrons. And each nephron is made of two distinct parts. One, the renal corpuscle. Okay, this is the area where filtration is going to take place. So that's around the glomerulus. Okay, it's literally it's almost like a baseball mitt that sits around the capillary bed of the glomerulus. Then the rest of our system is called the renal tubule. And we saw it in the last picture, right? It's literally just a tube that is coiled. It looks kind of like a mass of headphones after they've been in your pocket for too long. But that's where we are going to see our conversion of filtrate, things, aka what we filtered out of the blood, into urine. All right, so our renal corpuscle. Now, this consists of two things. Number one, the glomerulus. The glomerulus is capillary bed number one. All right, it's literally, it looks like a ball of vessels, okay, a ball of capillaries. Now, that ball of vessels is surrounded by a capsule, a tissue layer, specifically two tissue layers that are going to enclose that ball of capillaries, okay? So, let's look at the capsule itself. Two layers. Outside is called the parietal. The inside is called the visceral. Now, the outside, simple squamous epithelium. Nothing crazy. The inside, we get a unique kind of cell called a podocyte. Literally, it means foot cell. But we're going to see exactly why we call it that in the next picture. All right, so those podocytes wrap around the capillaries themselves, and they're going to form essentially a filter that's going to help keep the blood in the capillaries but remove the wastes. Now, between these two layers, we get the capsular space. So between the parietal and the visceral layer is a space made by these two layers of the capsule. Now that space will eventually become the lumen of our tubule. Now we also get different regions of this corpuscle, and the names aren't complicated. Okay, the vascular pole. Guess where it's going to be? It's going to be where the vessels enter and exit. The urinary pole. It's going to be the side where the tube begins, right, where the urine collection is going to begin. All right, then lastly, we have these mesangial cells. Again, another specialized type of cell here, and their job, okay, they pack the space among the capillaries. So they're kind of in that space between our parietal and visceral layer. And they are going to help regulate blood flow, and they can also phagocytize. So if any debris were to get in to the kidney, to the tubule portion, these mesangial cells can take care of it. So here is our renal corpuscle. Now, this side, what are we looking at? If you guessed the vascular pole, you would be correct, because this is where the blood enters and exits our capillary bed. That would make this end the, if you guessed urinary pole, you would be correct. This is where the filtrate, okay, will exit the capillary bed. So, outside layer right here, okay, this is our parietal layer. There's our simple squamous cells. We get the space, the capsular space in between. And then the innermost layer, that visceral layer, literally sits around the capillaries themselves. Okay, it kind of hugs them, and it's made up of those podocytes. which we'll see in more detail a little bit later. So what we're doing, we bring the blood in, the blood cells, the erythrocytes, stay in our capillary bed, but the wastes are filtered out through these podocytes. So they filter out, they enter that capsular space, and they will eventually drain into the tubes system, okay, the tubular system, where we will see more reabsorption, more excretion, and then at the end of this tubule, we will eventually have urine. All right, so what does it actually look like? The histology. A little bit harder to see, okay, but we are going to be seeing the same things here. So the big circle that we are seeing, that is your renal corpuscle, okay, this whole thing. The outermost layer of it, that would be the parietal layer. If you're seeing white, okay, that's empty space. So that's our capsular space, all of this. Now, the inside, this is a big old cluster of vessels. Now, these little red dots that you're seeing, okay, those guys right there, those are red blood cells. All right, so we're very, very zoomed in here. But those are individual red blood cells in these capillaries, and the lining of these tubes, all of these tubes, okay, would be the visceral layer of our capsule. Now, you can also see some tubes, okay, again, any white space. Okay, so this, for example, is part of the nephron loop, which, again, that's our loop of Henle. We're going to see that a little bit later on. We can see some proximal tubule here and here. Okay, again, these are just tubes. And then we can also see some of our arterioles and other capillary beds surrounding. So this is, okay, our tube here, the afferent arteriole, and you can see it's a tube, but this one's filled with red blood cells, and it's entering our glomerulus, so this would be the vascular pole coming in here. And then this one over here, again, it looks like a tube filled with red blood cells. This would be a capillary bed surrounding the glomerulus. So it's outside of our glomerulus. So this would be something like a peritubular capillary that we're seeing here. All right, so that is our renal corpuscle, okay, glomerulus and the capsule surrounding it. The second part of our nephron is called the renal tubule. Literally just a collecting duct, a series of tubes leading away from that glomerular capsule, and it's going to end right at the tip of the medullary pyramid. So it's going to be right at that papule. Now, we actually get 4 regions of this tube: the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and a collecting duct. So, proximal and distal. Proximal means close to, distal means away from. So, this is in terms of the glomerulus. We have the glomerulus, the renal capsule. Then we get the proximal convoluted tubule. So the closer tubule. Then we get the nephron loop, and that's this big guy that dips down that we talked about before. I called it the loop of Henle. All right, that's same name, same thing, nephron loop. Then we get the distal convoluted tubule. So if I could draw this out, here's our glomerulus. Then we get our tube system, going crazy. Ooh, convoluted tubule, loop of Henle. Distal convoluted tubule, ooh, tubes. And then we would drain into a collecting duct. Now, the proximal convoluted tubule, this guy right here, this is our longest and most coiled region. And what we're going to see, each area of this tubule is going to have different cells, okay, because each area has a different function. And of course, structure equals function. So we're going to see some similarities between them, but a lot of them are going to have unique structures. So again, proximal tubule, longest and most coiled. We get simple cuboidal epithelium with prominent microvilli. Now, let's think back to digestion, right? Where have we seen microvilli? Well, we saw microvilli in the intestines. And why did we see microvilli? What's the point? The point of our microvilli were to increase surface area, right, of our intestines. The bigger the table, the more hamburgers you can fit on it. So when you see microvilli, you should think absorption, right? If we're trying to increase surface area, I'll bet you everything I own that absorption is going to take place in that area. So structure equals function. Okay, simple cuboidal, prominent microvilli means we're going to be absorbing stuff in the proximal convoluted tubule. All right, I know you were going to miss my high quality drawing of the nephron, so I redrew it here for your viewing pleasure. Our next part of the renal tubule is called the loop of Henle, also called the nephron loop, which is this guy right here. All right, now again, we're going to see some differences based off of function. So when we're talking about the downward portion of the loop, guess what we call it? That's right, the descending loop. Okay. Then when we turn around and go back up the other direction, that is called the ascending loop. Now, if you look at these different regions, they are going to look different. They're going to have different cells. Okay, and I even kind of drew it on here, right? If you look at sometimes part of the descending limb, but most of the ascending limb typically is thicker. Okay, it's called a thick segment, and that's because of simple cuboidal epithelium. All right, if you look at the descending loop, so this guy here, the majority of it is thin segments, and they're thin because they have simple squamous epithelium. Now, structure equals function. Okay, when we're talking about the thick segments, and again, that's kind of just right at the initial part of this descending, and most of the ascending are thick segments. They are specialized to essentially transport certain things. Active transport of salts is big in this area, and you're going to see lots of mitochondria because active transport requires energy. So got to have lots of mitochondria in this area, hence why the cells are a little bit thicker. The thin segments, very basic simple squamous epithelium. This is the lower parts of the descending limb, and the reason why we want this cell type is because of its permeability. Simple squamous epithelium makes it very, very easy for water to move through. So the thick segments are for salts. Sodium is big. Active transport. The thin segments are for water. All right, so that's our loop of Henle, our nephron loop. Then we have the distal convoluted tubule. This region right here, highlighting it in blue. All right, so this is beginning right after the ascending limb re-enters the cortex. So what do I mean by that? I don't think I've actually mentioned it before. It depends on the nephron type that you are looking at, but if you are looking at, let's say, a juxtamedullary nephron, or even cortical nephron, the majority of the distal and proximal convoluted tubule sit in the cortex of your nephron, but this loop of Henle almost always dips down into the medulla. If we're talking a juxtamedullary, it will dip down very far into the medulla. If we were talking, let's say, a cortical nephron, it wouldn't dip down quite so far into the medulla, or sometimes it wouldn't dip into the medulla at all. So that's what it's referring to here, is when that ascending limb of the nephron loop reaches the cortex again. Now we're in the distal convoluted tubule. There we go, something like that. Now, cells in this area. Okay, the tube itself, the distal tubule, is shorter, it's less coiled, and we get specifically cuboidal epithelium. But we don't see nearly as much microvilli. So what does that tell us? We don't see as much microvilli. That means we're not going to see as much absorption. Less absorption will happen in this area because we don't care quite so much about surface area. All right, so we have reached the collecting duct. So we have passed the distal convoluted tubule, and now we are in this portion right here. And just like the name suggests, the collecting duct is going to receive the fluid from our tubule, okay, and it is going to collect it. It's going to collect that fluid and it's going to drain that fluid out of the kidney. So I've collected my filtrate here, and the collecting duct is going to serve several nephrons. So each of these little red lines that I drew would connect back to different nephrons, different units, and all of them would drain the fluid into this distal tubule. So numerous collecting ducts, or numerous nephrons will dump into the collecting duct. Then as you follow it further down, okay, multiple collecting ducts will converge toward the tip of that medullary pyramid. So we're right at that papillary region, the papillary duct, right at the tip of, remember, our little medullary pyramid. Okay, here's the cortex up here. Pyramid. So all of this is happening, all of this is draining toward this region, and then these little papillary ducts at the tip of each pyramid are going to collect that filtrate, and they are going to drain it into a minor calyx. Now, tissue in this area, tissue type, it is just simple cuboidal epithelium in the collecting and papillary ducts. We are going to see slight reabsorption occasionally in this area, but at this point it's usually more about collection than it is about reabsorption. So here is our flow. We start with the glomerular capsule, proximal convoluted tubule, nephron loop, distal convoluted tubule, collecting duct, papillary duct, and then we begin the true draining system. Minor calyx, major calyx, pelvis, ureter, urinary bladder, and urethra. All right, so this is our full flow of fluid, of excretion. All right, here's a much better picture than mine that I drew to help you guys get an idea. So again, we start with the glomerulus in its capsule. Okay, we filter out the wastes from the blood. The blood continues out through the efferent arteriole, but our filtrate, our fluid, begins the journey through the tubule system. So proximal convoluted tubule, we change a little bit. We see some thick segments in the upper portion of our nephron loop. Thin segment here, water reabsorption here. This is mostly sodium absorption. Again, we'll see a lot more on that later on. Then we turn, we get our thick region of the ascending loop. Our distal convoluted tubule drains into the collecting duct. All this guy here, multiple collecting ducts drain into a papillary duct. which drains into a minor calyx. All right, and again, here's this renal papilla and the minor calyx that will collect all of that filtrate, which we can now officially call urine once we've reached this area. So again, this is the full nephron, but again, we can classify our nephrons based on their kind of positioning. So cortical nephrons, which are something like this guy here and this guy, they are going to sit pretty much entirely within the cortex, or they might dip a little bit into the medulla. But juxtamedullary nephrons, this guy right here, this guy sits in the cortex. Okay, so the glomerulus and the coiled tubules sit in the cortex, but this loop of Henle sits almost entirely in the medulla. All right, so that's how you tell the difference between these two nephron types, is look for that loop of Henle and see how far it extends into the medulla. So this would be a juxtamedullary, what I just drew here. And then the cortical would be something like this, where it maybe just barely dips into the medulla, or, and here this is our cortical, it would just barely dip into the medulla, or sometimes the medulla is all the way down here and it doesn't even cross into the medulla. So that's our difference. Look for how far that loop of Henle dips down. All right, so here are the official descriptions of our two types of nephrons. Juxtamedullary, we are close to that medulla, very long loops of Henle that will sometimes dip all the way into the apex of the renal pyramid. So very, very long loop of Henle extends very far into that medulla. Only 15% of your nephrons are juxtamedullary, but they are very, very, very important for the osmotic gradient in your medulla. So again, we're going to talk about this more later on, but when it comes to absorption and the movement of water, these guys are going to be key players. Okay, if you need to conserve water, juxtamedullary nephrons are going to help with that. Cortical nephrons, much shorter loop of Henle. Okay, they maybe dip only a short way into that medulla. Sometimes they don't even have a nephron loop at all. So very, very different composition here. And the majority of your nephrons are going to be cortical nephrons. So before we really get into the details, we need to talk about the nerves, okay, renal innervation. Now, there is a plexus called the renal plexus, which again, just a clustering of nerves and ganglia, which are going to essentially cluster around each of the renal arteries. Then it's going to follow that renal artery in at that hilum region into the parenchyma, so the tissue of the kidneys itself. Now, essentially, it's going to follow the blood vessels. Okay, the nerve fibers are going to follow the blood vessels and surround the convoluted tubules of the nephron. Now, sympathetic innervation here is going to be important. This is coming from the abdominal plexus, the abdominal aortic plexus. And whenever you stimulate the sympathetic, we are going to reduce glomerular blood flow and reduce the rate of production, okay, urine production. Again, when you think sympathetic, you should think fight, flight, or as we'll learn soon, fornicate. But for parasympathetic, that's your rest and digest. So creation of urine and urination is part of that rest and digest. So it's going to be stimulated by the parasympathetic. Sympathetic will reduce, okay, reduce production. Now, we also see that we can kind of control some of that output. We can send signals to our kidneys through this sympathetic innervation in response to falling blood pressure. right? So remember, when it comes to blood pressure, blood volume plays a big role, and blood volume is all about how much water you have in your blood. So all you have to do, let's say your blood pressure falls, you can stimulate the kidneys to secrete renin, which is going to help activate certain mechanisms to restore blood pressure and help the kidneys retain more of that water in our blood. So we're going to talk more about that later, but onward, parasympathetic. Okay, we do see parasympathetic innervation. However, we are still kind of unsure of the exact function. For a long time, it was believed that only the sympathetic nervous system had control over the kidneys. But we more recently found that yes, there is parasympathetic innervation, which would make sense with the rest and digest. So there's some ideas on it. But we still don't know exactly how the parasympathetic nervous system stimulates this production of urine or the flow to the glomerulus. So this part is still relatively new to our knowledge and relatively unknown. But we do know sympathetic is going to reduce. All right, so how do we do it? How do we actually make urine? Okay. We're going to talk about it, and this is one of the more complex areas of the kidney. We're going to see a lot of osmosis, we're going to see a lot of diffusion, active transport, so it's really important to understand how things like to move. So I've written some reminders up here for you. I didn't feel like writing it all, so this first one is diffusion. Okay. Diffusion is the simple movement of molecules, and they like to move from high to low concentrations. If you ever wanted to reverse this and force something to go from low to high, we would need energy. We would need some ATP. This is active transport. All right. Osmosis, also important here. Osmosis likes to move from low solute to high solute. So water likes to go be with other things. And we saw that a little bit when we talked absorption in the intestines, right? Because if you bring in, let's say sodium, for example, or glucose, water will follow, right? It likes to move wherever there is more solute, more stuff. So keep this in mind as we go through. We're going to start with step 1, glomerular filtration. All right, the first thing that we have to do, we bring blood into the kidneys, okay, and it's going to go directly to that glomerulus, right? It's going to go into smaller and smaller arteries, and then we bring it to our first capillary bed. All right, so this is stage 1, glomerular filtration, and you'll see there's 4 distinct stages. Now, we give this fluid different names as it passes through the nephron. All right, when we are in the nephron, you will typically call the fluid filtrate or fluid. If it's in the glomerulus, it's filtrate. If it's in a tube, it's tubular fluid. The only time we can officially call it urine is once it has made it through pretty much the entirety of our nephron, collecting duct and beyond. All right, so be specific with your naming here. All right, so here are the basic stages. Number 1, glomerular filtration. Bring the blood in, we hit our first capillary bed, and we filter it. Okay, all of the wastes will make their way into this space between our layers, the parietal and visceral, and will drain that filtrate into our tube, okay, this way. Now, that begins step 2, tubular reabsorption. All right, we need to make sure we don't get rid of anything useful. So a lot of times we will see reabsorption, okay, bringing back sodium, okay, bring back essential ions, things that we don't want to get rid of, but things that got accidentally filtered out. So, reabsorption. Step 3, tubular secretion. All right, we need to make sure that we get rid of all the waste. So maybe it didn't make it out in our first capillary bed. Well, guess what? We still have another option. We have a second capillary bed. For example, the paratubular, right, that are going to sit around the tubes themselves. And we can secrete some stuff from that second capillary bed, added in more wastes we can shove into that tubular fluid. Lastly, water conservation. We need to make sure that we are keeping and discarding the appropriate amounts of water. So, for example, let's say you're dehydrated, you don't have a lot of water. We would want to concentrate that fluid. We would want to remove and retain, keep as much water as we possibly can, because we need it. All right, our urine would be very concentrated. However, opposite is also true. If you are overhydrated, right, you will instead push more water into the fluid, okay? Push more water into the fluid, and that will drain out into the collecting ducts. So we can essentially conserve or get rid of water based off of the needs of the body. Now, once we hit the collecting duct, we can officially call it urine, and it is on its way out. All right, so we're going to start with glomerular filtration. So we have to understand what's going on at this first capillary bed. All right, we are trying to filter out the wastes. And how we do that is with a filtration membrane. All right, think of coffee in a coffee filter. Right, if you put coffee in a coffee filter and you pour water through it, okay, the water can get through, but the coffee grounds are too big to make it through the filter. That is the same idea here. We are going to be passing the fluid from our blood through a filter. Okay? Certain things are too big to get through, like our red blood cells, for example. We keep those. But the wastes can get filtered out. Unfortunately, water can also get filtered out, and a lot of solutes, like sodium, for example, can also get filtered out. So the wastes, the water, the solutes are all going to pass from the blood within the capillary, the glomerulus, into the capsular space of the nephron. So let's talk that membrane. What does our coffee filter actually look like? Well, there are 3 layers to this membrane, 3 filtration layers. The first is the capillary itself. Okay, the glomerular capillaries are fenestrated capillaries, meaning they have large pores. Now, this isn't a sinusoid. It's not big enough that actual blood cells can pass through, but these fenestrations are big. Okay, 70 to 90 nanometers, which is still highly permeable. So this is going to, a lot of stuff is going to be able to get through. All right, right after that, we have what's called the basement membrane. Now, this is essentially a proteoglycan gel, and it has a negative charge, which is very helpful for us. Because remember, with charges, opposites attract and like charges repel. So if you have a membrane with a negative charge, it's going to repel anything else with a negative charge. So a lot of the smaller molecules, things that are going to be greater than 8 nanometers, pretty small, but things like albumin are going to be repelled by that negative charge, and that's what we want. We need to keep albumin in our blood in order to maintain its osmolarity. All right, the last component of our membrane is called the filtration slits. And finally, you'll get to know why we call it a podocyte. Okay, podo means foot, cyte is cell. And the reason we call it this, these little cells wrap around the capillaries and they have little feet little pedicels, foot processes, that it will essentially sit around the capillaries themselves, and they will form another membrane, another filtration net to get through. So between these foot-like processes are the fenestration slits, the last membrane, the last element of the membrane that our filtrate would need to pass through to officially make it into the capsular space. So this is the direction that we're going. Okay, if you are in the blood, you would have to make it through a fenestrated capillary. You would then have to make it through the basement membrane and the fenestration slits in order to enter the capsular space. Okay, and this would be from the blood. So let's see what that actually looks like. On the left over here, this is the glomerulus. You can see the afferent arteriole bringing blood in. All of these little ramen-like tubes are the capillary bed of the glomerulus, surrounded by that visceral layer. And then here is the efferent arteriole bringing blood out. Now, over on the right, we've zoomed in even more, and this is to show you a podocyte. So this big blue guy right here is a podocyte, and you can see its pedicels, its extensions, these little feet that it is extending onto this capillary, and it makes these little fenestrations, these little narrow slits that are again going to repel and essentially serve as a filter, okay, keeping a lot of the stuff we want to stay in to the blood in. And then the filtrate is able to make it through that filter into the capsule space. Here is another view where we can see it a little bit better. So this area right here, this is all vessel, okay, capillary. You can see a erythrocyte, this big dark dot right here. Then we can see the endothelium, okay, right here, endothelial cell, okay, of our capillary bed. Okay, so that endothelium and the fenestrations are your membrane number one, the first part of your coffee filter. We get some fenestration slits. We get some filtration pores that are going to help things move through. And then here is our capsular space. This is what we're trying to get fluid, the wastes, into at the end of the day. And this big guy over here is your podocyte, and you can see his feet extending and making these little fenestration slits. So how much can we filter out? Well, if you are smaller than 3 nanometers, you can freely make it through the filtration membrane. So a lot of stuff is smaller than 3 nanometers. Water, electrolytes, that's your sodium, chloride, potassium, glucose, fatty acids, amino acids, wastes, vitamins. All of these can make it through that filtration membrane. And some of this is good, right? We do want our nitrogenous wastes to make it out, but we don't necessarily want glucose or electrolytes or water making it out. Just depends on the needs of the body. So that's where the reabsorption comes back in. Okay, we need to bring some of that stuff back into the blood a little bit later on. Now, if you are a substance that is below 3 nanometers, have a low molecular weight, if you are bound to a plasma protein, certain large proteins, you might not make it through the membrane. So a lot of your calcium, iron, and thyroid hormone will be kept in your blood because they're bound to specific plasma proteins that are too big. However, if you have unbound calcium, iron, or thyroid hormone, they can pass freely into the filtrate. So the last thing I want to talk about is how can we use the composition of filtrate and urine to our advantage, right? Certain things, for example, kidney infections or trauma, any damage to that membrane, that filtration membrane, can allow things that aren't supposed to get into your urine get into your urine. All right, so for example, if you have kidney damage, if you got punched in the kidney and it damaged some of those filtration membranes, you might see something like hematuria. Literally, hemato referring to blood, urea referring to urine. You have blood in your urine. Now, red blood cells are supposed to be too big to pass into that filtrate, to pass that filtration membrane. However, again, you damage the membrane, you poke a big hole in your coffee filter, right? You're going to see some coffee grounds making it through. Now, same thing, kind of separate. Proteinuria is literally protein in the urine. This is when we see the big protein albumin in your urine. And again, this can indicate issues with the kidney. All right, so this is our last slide of the video showing you the filtration membrane and what can get through. So, first layer, we have the endothelium of our capillary bed, and you'll see it is fenestrated, meaning it has these fenestrations, these gaps that things can get through. Then we have in blue in the middle our basement membrane, and on the other side, these guys are the feet of the podocytes, so the pedicels of the podocytes, and how they come down makes these little filtration slits that things can move through. So these are the barriers. The big stuff that's turned back, that's your blood, plasma proteins like albumin because of that negative charge on the basement membrane, large anions, anything that's protein bound, so like protein bound calcium, for example, anything that's greater than 8 nanometers. The stuff that can get through, water, electrolytes, that's ions, again, sodium, for example, calcium that's not bound to a protein. Glucose can get through, amino acids, fatty acids, vitamins, waste products. And again, the waste products are what we want to get out. Some of these, we might have to reabsorb. We don't want to lose too many electrolytes or too much water. But we will see that in the next step of our urine production.